And we've got it. All right. Welcome, everybody. We're just going to give it a second here for the rest of you to get off of work and join us. And let me get my screen up over here. Okay, we got 71 joined already, 76 total. Okay, we're moving along here. All right, everybody. Welcome to the HCMA's Bighearted Warrior Tour Special Edition. And tonight we are going to be bringing you some really interesting topic. Mostly we're going to be talking about gene therapy in hypertrophic cardiomyopathy, an exciting and really courageous move forward in medicine. But it is a trial we're going to be talking about tonight. I will be reading a disclaimer shortly. Before I get started with my presentation today, I want to introduce our speakers and our faculty and our staff. First, I'll start with the HCMA staff. I want to thank Stacy Titus and Julie Russo for all their organization of the Bighearted Warrior Tours. And if you have any technical difficulties during the evening, they will be in chat there to help you. We're going to ask that you use the Q&A box for questions, not the chat box. And they will be monitoring the chat. If there's any technical issues, I will be monitoring the Q&A and moderating questions as we move forward. If I didn't introduce myself, I'm Lisa Selberg, the founder and CEO of the HCMA. And every once in a while, we're really, well, all right, maybe like once a month or so, we're really lucky to have some amazing clinicians, researchers with us to share information. And today I'm going to do ladies first. Luisa Mastroni, which I hope I pronounced that right, who is with us from the University of Colorado. Luisa, could you please tell the audience a little bit about your background, because they may not all be familiar with you.
Thank you, Lisa, for the introduction. I am a professor of medicine and cardiology here at the University of Colorado Anschutz Medical Campus. I direct the Inherited Cardiomyopathy Clinic, and I'm the co-director of the Molecular Genetic Program of the Cardiovascular Institute. I have been in this field for decades. And my research in particular goes from clinical studies, outcome studies, risk prediction models to stem cell models, and more recently, very much into new therapies. And this is what we will talk about today.
Fantastic. Thank you so much. You give your bio so quickly. It's a lot to absorb, but you've done some amazing work, and we really appreciate that. Hold on one second. I have a technical issue, I think, on my... You're not seeing Facebook Live. It's there. Hold on one second. We may have a technical difficulty. Hold that thought. Okay. Me and my Facebook thing are getting a little twitchy at the moment. Here we go. You're not seeing it. I'm seeing it. Sorry, guys. Technology. I am live. Ross just checked in. Something's wrong with Stacy's computer. Always good to double check. Joe Rosano is coming to us from CHOP, and maybe not familiar to a lot of our adult HCM families, but to those with littles, they probably know Joe. Tell us a little bit about your practice.
Yeah, hi, Lisa. Thank you so much. It's a real pleasure and honor to be here to be able to speak with you tonight. So yeah, I'm a pediatric cardiologist, and I'm the chief of cardiology at the Children's Hospital of Philadelphia, where for a pediatric, size program, have a fairly large cardiomyopathy program. And my clinical and research interest has been in cardiomyopathy and heart failure in children, and have been involved with research in this population throughout my career. And pertinent for tonight's talk, I am our site PI for a gene therapy trial for Danon disease, and we'll talk to you a little bit about the experience and what we've learned so far from that study.
Fantastic. Now I'm hoping that some of our participants are saying, who are these people and why are they talking to us about this? Have a better understanding of why I'm so excited to have them here tonight. Okay, I am going to do a quick presentation. I'm going to take about 10 minutes to update everybody a little bit about what's going on here at the HCMA. So I'm going to do oops, I have to undo that because you need to hear my audio on this one. Share sound, because we have a little video we're going to show you. Okay. So again, welcome to everybody to the Bighearted Warrior Tour Special Edition Gene Therapy. And we are really excited to be talking to you tonight about the basics of Gene Therapy 101, what we've learned from prior AAV9 cardiac therapies, and the role of immunosuppression in gene therapy. We're going to take all your questions and hopefully get you some good answers. I am going to launch the poll. And if you could take a few minutes while I'm jammering on and take the poll, I'd really appreciate it. It lets us know where you're coming to us from and what some of the issues are that you're thinking about today. We want to thank our sponsors of this particular webinar, which would be Tenaya Therapeutics and BioMarin. And you will be getting a survey after tonight's presentation. And we're trying to do a few things with that survey after the event. Number one, you'll be able to give us some feedback on what you learned about gene therapy. Number two, you'll be able to inquire directly to one of the sponsors of a clinical trial about that clinical trial. So you will be hearing back from us a little bit differently than you typically hear back from us after a webinar. So that being said, I'm going to read a disclaimer for this webinar, and I'm going to read it into the record. So this webinar is intended solely to provide information. The information presented as part of the webinar and any materials published in relation to this webinar are provided for general purposes only and should not be construed as professional advice from any of the hosts, presenters, sponsors of the webinar, or any of their respective affiliates, associates, employees, or representatives, collectively the presenters. The content of this webinar may not directly apply to certain circumstances. Professional advice should be sought before any action is taken in relation to the information disseminated during this webinar. Genetic therapies for HCM are strictly investigational at this time, and we are currently in July 2023. In case anybody sees this later. So one of the missions of the HCMA is to help to find the undiagnosed. And one of the things that we are doing here at HCMA to find them is moving forward on a legislative initiative called the Healthy Cardiac Monitoring Act. This is a state-based legislative initiative to include all child examinations to include cardiac components, just like we do for student athletes. This would make sure that we are doing the student athlete pre-participation screening physicals, as well as checking all families for family histories of genetic cardiac disease. We seek to develop professional development tools for the providers in the states where we pass this law. And we are taking this approach because we find it sustainable, reproducible, and cost-effective. We've been doing it in New Jersey since 2015, and this is going very well. Referrals are being made, diagnoses are being made, and families are being treated. So you're going to hear more about this over the coming days. And what does it include? It is adding well-child exam inclusion of those 14-point screening for heart disease in every well-child exam, ages 1 to 19. We're going to develop professional development tools like we have for the state of New Jersey and put those out nationally. We're going to improve the student athlete participation physicals by making sure that they are standardized. We want to improve healthcare providers' ability to identify children and families at risk for cardiac disorders, both genetic and congenital. And while it may not always come under the name of the Healthy Cardiac Monitoring Act, some states have pieces of legislation that are doing well, but we want to fortify them with the elements of the Healthy Cardiac Monitoring Act. And then the next few weeks, you're going to see a new feature from the HCMA, and that is going to be a map of the United States. You will be able to click on that map, and it will tell you how well your state is doing in cardiac safety and patient access. So you'll be able to see how you're doing on AED placement, CPR training, and other factors related to sudden cardiac arrest identification and response, as well as issues related to medication management and access, including step therapy laws. Okay, I'm not sure what that's about, as well as making sure that there is a deep understanding in the medical field about who should be referred for cardiac evaluations going forward. I want to take a moment here to talk about how to find active clinical trials on the HCMA website. If you go to the HCMA website, and I know this has a couple of links here, you're going to go to the main page and we have the HCMA journey, and then you'll see the research journey. And there's a field called antidote, and that brings you to a clinical trial finder. All you need to do is follow along on this. Whoop, sorry, I thought this clicked through. Follow through on the little green buttons, and it will attempt to populate with whatever you're looking for. So we have this looking for hypertrophic cardiomyopathy trials. And if you put in your age, your zip code, you do not have to put in personal identifiers in terms of an email or a name. You can just search to see what's available. You will find a lot of clinical trial opportunities right through this tool. And we are hoping that at the end of tonight, you might be encouraged to take a look at some new clinical trials that might be available. We want to remind you that if you are trying to get to HCMA Center of Excellence care, but are having financial difficulties, that the HCMA is here to help you. We will be providing up to $600 in travel expenses per year for a family to go to an HCM Center of Excellence. You must meet some guidelines on the federal poverty guidelines, and there are some documents you will need to complete. But we were happy to say that we have completed at least a dozen at this point, I believe, Lori Fund grant recipients. And we're happy to say that it helped families get to the care that they needed. So you can apply at any time, or you can donate to the program as well. My last few minutes, I'm going to spend on something not related to gene therapy. I will have a couple of other items for you at the end of the session tonight. But we've been talking about Drill Dr. Heart for most of 2023. We started the year with a very public cardiac arrest. We had another one last week, and they don't have sudden cardiac arrest is a common way for people to unfortunately collapse, and hopefully we get them back. So HCMA has a project called Drill Dr. Heart, which we've had going on since about 2009, but we've revamped it for the modern era, and we're going to present to you a video on how you can drill for a cardiac emergency in your workplace, in your school, at your team, anywhere you go, anywhere you're working, anywhere people gather, you can do this drill. We're showing it in a business setting, but you can show it anywhere. Okay, this is the HCMA's Drill Dr. Heart drill. Enjoy for the next 3 minutes. So now we've completed all of our planning for HCM Awareness Day. We need to do our annual Drill Dr. Heart training. So we need to make sure that everybody knows what to do in the event of a cardiac emergency here at the HCMA. So as you know, the AED is stored right by the front door on the entry table. So if anybody needs it, that's where it is. And I do make sure that the batteries are charged and the pads are fresh by checking the battery indicator light frequently. Now, if there's a cardiac arrest, what are we going to be doing? We're going to be calling 911. We're going to be starting compressions as soon as possible. We're going to be bringing the AED to the point where the person has gone down. We are going to make sure that EMS knows how to enter the building. And if there's enough of us here, somebody's going to go down and meet EMS and bring them up, because we know our building can be really confusing to find us. And how many here have CPR certification? Raise your hand. Okay, so everybody's CPR certified. Yay. Always good to have us all ready to respond. Now, has everybody gone over AED training in your CPR training as well, and you know how to use the device? So are there any questions about the steps that are involved in providing life-saving care should one of us have a cardiac arrest? No questions? We've got this?
Ready.
Are you ready?
Oh, I'm so ready.
Everybody's ready. So we have done our discussion. We know the roles. We know who's CPR trained. We understand what we need to do. Let's hope we never need it. Great meeting, guys. Thanks.
That was last week?
Yeah, July. You believe it's already July?
I believe that actually.
Oh, morning, Lynn.
Surprise.
Hey. Oh,
Morning.
hey, you okay?
Okay.
Oh my
Oh,
God.
Olivia. Oh, are you okay? Drill Dr. Hart, everybody. Olivia's down.
I got 911.
I got the defibrillator. Okay, I'll go get EMS. Let's clear this way.
Yes, possible cardiac arrest. 66 Ford Road.
Room
Second
closed from
floor,
patient's
suite
chest.
213B.
Peel
Yes,
right.
possible cardiac arrest.
Stay clear of patient.
Thank you.
All right, let's stand back.
Said a
Analyzing
few
heart
minutes.
rhythm. Stay clear of patient. Analyzing heart rhythm. Shock advised. Stay clear of patient.
All right,
Press
everybody
the
stand back.
flashing orange button now.
Pressing the button.
Shock delivered. Be sure emergency medical services have been called.
The neighbor's
It is
waiting at
safe
the door
to
for
touch
EMS.
the
How's she
patient.
doing?
Great.
Begin CPR. For help with CPR, press the flashing blue button.
Okay, team, that's how you do a drill, Dr. Hart. Well done. That is how you do a drill, Dr. Hart. And if we can get more workplaces, teams, schools, and community organizations to drill for a cardiac emergency, we can ensure that they are all prepared for such emergencies in the future and hopefully save more lives. So you can learn more about that on the website. Oh, there we go. We want you to take the challenge, plan for the drill, practice it, film it, and post it, and your community event or organization, can win a free AED. Please visit the website to learn more. We also want to notify you that on October 21st, we're back for a live meeting with real people. You can see each other's shoes and everything. It will be a day of learning, a day of education. It will be a CME course for those of you who need CME credits. We're going to have a very jam-packed 3-room agenda, and we're going to talk about shared decision-making, health equity, global patient advocacy. We're going to talk about myosin inhibitors. We're going to talk about gene therapies. We're going to talk about lots of late-breaking information and clinical trials in HCM. And at night, we're going to celebrate. We're going to celebrate some centers, some volunteers, and a very special person in the HCM history, Mr. Michael Moss. He is a Pulitzer Prize winning New York Times bestselling author, and he has some really interesting ties to the beginning of the HCMA. You're going to hear all about it at the event, and we're really excited to have him there. Additionally, we're going to, my slides were a little out of order. That's where I was confused. That antidote clinical trial finder that we addressed earlier in my talk, these are the key studies that you're going to find tonight. We're going to be talking mostly about the first one here on the list, and that is a clinical trial brought to you by Tenaya Therapeutics, the study on the safety and tolerability of TN-201, adults with symptomatic myosin-binding protein C mutation-associated hypertrophic cardiomyopathy. There are other really interesting clinical trials going on as well. You can find them all on the Clinical Trial Finder at 4hcm.org under the My Journey, My Research, Antidote. Okay, I want to say thank you to our board, our staff, our volunteers, our partners, and our faculty for tonight's presentation. And of course, as always, thank you to Brandy, my donor, for her. Without her, I would not be with you today. So I'm going to end my presentation there. I hope you enjoyed our presentation of Drill Dr. Hart. And before we get started with, I'm sorry, who's going first? I keep doing this. Luis is going first. I keep going backwards. Probably should keep a copy of the agenda in front of me. Let's take a quick look at who is here with us. We have 71% participation right now. Most are the Northeast. You win by 37%, but we have the whole country well populated and Canada and other. We have 4 others. If you're an other, please send us a note in chat what other country you're coming from. What relationship do you have to HCM? 52% are patients, 18% are patient and family member. You know what? I can show you guys this too as I'm doing this. That's where you're all from. Okay. 18% are a family member of somebody with HCM. We have 3% medical providers, 9% industry, and 1% just curious. Just curious. You always be curious. Have you had any of the following? 9% of you are diagnosed in the last 2 years, 63% on meds, 47% have had an ICD, 25% have had a septal reduction therapy, 30% experience AFib, 23% of us have lost somebody to HCM, 3% are transplants, so that makes 3 of us here. This is nice. 65% of participants tonight have had genetic testing for HCM. We may have just set a record there with the most percentage of patients being genetically screened in one place at a time. 5% have been advised to have an ICD or but are considering. 4% have been considering septal reduction therapy. And 16% are evaluating treatment options at this time, and 65% are being seen at a center of excellence. Okay, so we've got a potpourri tonight. We've got new and old, but we have 65% who have had gene testing already. That's pretty exciting. Luisa, it is off to you. Tell us about Gene Therapy 101.
It will be my pleasure. Let me share the screen.
Thank you.
Can you see my screen?
Perfect.
Great. So the idea to talk about gene therapy probably was a little bit prompted by a short presentation, a short discussion we had a couple of months ago about what is really novel in the field and what could be very important also for the patient to understand a little bit where the clinical research is going and what to expect in the next few years or few decades. So, and I apologize if some of the slides are a little bit too basic or too simple for the audience that may be actually very highly educated in this regard. But this is a scheme from the NIH showing what is the meaning of gene therapy. So in the gene therapy, what you want to do is to, I think you can see my arrow, to replace a defective gene that is causing the disease in a patient with a healthy and functional and normal gene that is packaged, in this case, into a viral vector. The viral vector allows the entrance of the normal genes into the cell and into the nucleus, and at that point, the normal functional genes start working and, let's say, treat the disease of the patient. So this is the concept. So one of the important things to consider is that there are, so this seems to be futuristic in the field of cardiology and in particular for our patient. But the truth is that actually gene therapy programs and therapies has been ongoing in the world for a long time. So for over 15 years, there are thousands of patients that have been treated for a variety of diseases. There are 8 FDA-approved gene therapies. Of these, 5 actually use what is called the AAV gene vector. So for our purpose, so for the purpose of the heart disease, we will be talking about AAV9, which is the adenoviral virus type 9. And as you can see, there are a number of gene therapy trials that are occurring in the world, particularly in the United States and in Europe. So this is really not a complete novelty, even though it's a novelty for us. So I already showed a couple of months ago this slide, but because we have a little bit more time and the need to understand, let me go take you through what is the concept of gene therapy. So there are different types of gene therapy. One is the gene replacement therapy. So what is available now for, in particular, hypertrophic cardiomyopathy is this type of gene therapy. So what happened in this case, one gene due to a mutation is not working. So there is a loss of production of the a functional protein that is encoded by the DNA in this patient, by the particular gene. In this case, the normal DNA is packed into the gene. It's then translated into the normal protein, and this allows the rescue of the amount of the functional normal non-mutant protein level. So this works when the cause of the disease is what is called a loss of function. So there is a loss of the proper amount of the protein in the heart. And another approach is instead to silence the abnormal gene. The way it's done is the gene produce a RNA, which is called the RNA messenger, to produce then the protein. And using silencing RNAs that enter into the cell, we can degrade the abnormal mutant RNA, and so destroy the production of the abnormal protein. So this is also used in cardiology. By the way, very recently, I think last week, the New England Journal of Medicine, which is the major journal for us, published a study of using this technology for hypertension, so a very common disease. So this could have a significant impact, but has not been particularly successful or used in the case of hypertrophic cardiomyopathy. One of the problems that this type of approach has is that this requires several treatments. And then there is the ultimate possibility, which is actually to repair the mutation. So to correct directly into the DNA the mutant gene, causing then the defective protein. This is done using a discovery, the CRISPR-Cas technology, that it's able to repair the DNA in the target cells. We will discuss a little bit more in detail this. This is, of course, the ultimate and more exciting possibility, allows a precision correction of the mutation, but of course has also more challenges. So there are a number of gene therapy trials, and this is only a partial list that involve cardiac disease, from heart failure, coronary artery disease, but for what is our interest, Danon disease, that will be discussed by Dr. Rossano, which was probably the very first gene trial that we had in cardiac disease using a AAV vector. And then we will discuss about the Tenaya Therapeutics trial that is that involves hypertrophic cardiomyopathy caused by the myosin binding protein C mutation. Another trial is another company that is working in this regard is BioMarin, and they are both actually supporting this session. And there are other cardiomyopathies that now currently has been the focus of gene therapy. So this is something that it's happening, and it's good to know what we are talking about. So just the basic of the concept of gene mutation, the gene therapy in hypertrophic cardiomyopathy. So imagine a functional gene encoded by a normal DNA. it will produce a normal protein. The protein will be expressed in the heart muscle as it should be, and the heart function properly. But if there is a mutation that disrupt the production of the protein, there will be an abnormal quantity, an abnormal amount. This will impair the heart function, and this will cause the disease. So this is how we explain that a gene mutation can cause a loss of the normal amount of a protein, and this way cause heart disease. So the purpose of gene therapy is to treat or prevent the disease in our case. The way to do it is to insert a working gene into the heart muscle cells, and therefore restoring the normal production of the normal protein, and correcting the underlying cause of the genetic condition. And what is a frequent question that comes out, but is this something that then the patient that undergo gene therapy can transmit to the offspring or not? And the answer is not. So this is really a therapy that will involve only the organ of our patient, but it will not be transmitted to the future generations. So some basic about how this therapy is done. So basically you take an adeno-associated virus. These viruses are actually very common. They occur naturally. They usually don't cause significant symptom of disease. They are used because they are very efficient, and there are very specific subtype of the viruses that are targeting very specifically one organ. And in the case of the heart is the type 9. And how are they used? The viral genome is, of course, removed. You don't want to inject a virus. The vector is used, and inside the vector, the working gene is inserted. So what is done is that the terminology is that the normal gene, it's packaged into the viral vector. The normal gene contains also a promoter, which is basically a switch on of the gene, so that when it's delivered, it will actually be expressed and produce the normal protein when it reaches the heart. So how is this done? So as I said, so the viral DNA is removed, and a normal gene that is of interest is inserted into the viral vector. It's packaged, as we say. And the interesting thing is that it's administered only one time. So imagine that this is a little bit like a small transplant, so if I can say so. So it's actually an important procedure, as we will see, that requires special caution and during the period of the treatment. But basically it's done one time. And then once is in circulation, the vector and the normal gene targets the heart muscle cells, and start producing the normal protein and restores the function of the heart. So as Lisa already mentioned, so one of the gene therapy trials that have been approved by the FDA, and it's going to start very soon, is the gene therapy trial TN-201 of Tenaya Therapeutics. So the reason for, this is also an important thing to consider. So the reason to target myosin binding protein C3 is based on a number of factors, but one of these, so one is certainly that it's one of the major, the most frequent gene in hypertrophic cardiomyopathy. The other point is that it's a relatively small gene, so it can be handled by this type of gene therapy technology. The target is, of course, heart muscle cell. The problem is a patient that have a mutation and have hypertrophic cardiomyopathy because they are not able to produce enough of the normal protein. And the goal is to actually improve the heart function. So this is just a summary, so you have an idea of the protocol, which is currently the trial is in a phase, what is called a phase 1B study, so small number of patients, to evaluate safety and tolerability of the single intravenous infusion of the adenoviral vector. And it's in non-obstructive hypertrophic cardiomyopathy. There are two actually trials. One it's only in adult population, the other it's in the pediatric population. The patient eligible are symptomatic, and again, they have non-obstructive cardiomyopathy. They also must have a cardiac defibrillator implanted for safety reason. So this is happening, and we will discuss what are the question and the question that patients have and the physician have in regards of these novel therapies. Let me just spend a couple of words about the, as I said, the precision correction of the mutation that cause hypertrophic cardiomyopathy. What is called base editing, and show you some of the very preliminary preclinical, how they are called data. So as I said, the direct genome editing means that you basically take the normal DNA, take out the mutant DNA and replace it. This is a very intuitive image that I took from the Scientist journal. This is a little bit more elaborated, and this is taken from Wikipedia, to tell you that the way it works is that you have to have a special protein called Cas9, and then a methods that is based on a guide RNA. The guide RNA contains the correct sequence to be replaced, and very simply, a CRISPR-Cas9 package is like a mechanic. So remove the abnormal part, replace it with the normal part, and completely repair the DNA of the patient. This is on your right is a scheme that is instead taken from a scientific journal, and it's a little bit more elaborated, but it gives all the details of how this system that actually led to a Nobel Prize works. And the two options that are possible, so one is simply to cut out the gene. The other is to actually, what we discuss is the homology repair, which is the substitution of the mutant gene with the normal gene. So let's see what are the preliminary data that are relevant for hypertrophic cardiomyopathy. Well, two major laboratories. There's the laboratory of Eric Olson, and the other is the laboratory of Cricket and John Seidman. One it's in Dallas, Texas, and the other it's in Harvard. Were able to show encouraging results of this technology. Why this is important? Because some of the genes that are causing hypertrophic cardiomyopathy are too big to allow the replacement therapy. And the other aspect is that sometimes the problem is not that there is a loss of a protein due to the gene mutation. Actually, the amount of the protein is normal, but the protein, like the gene, has a mutation that make the protein not work properly. And this is the case of beta-myosin heavy chain gene. a very large gene, frequently with one single base mutation, like the case we are looking here. This is actually a mouse model that is used, and it's very important for preclinical studies before going to man, to the human subjects. And these are the results of the gene therapy in the mice. So this is the normal heart. This is the mice with the 403 myosin mutation. It is a very well-established mutation causing hypertrophic cardiomyopathy. As you can see, there is a striking difference. So first of all, this is the thickness of the heart muscle. As you can see, the thickness is definitely less in the normal heart. The other thing is that the space between the two walls, which is contained in here, it's much smaller here because of the hypertrophy. And this on your right is the mice after gene therapy. And as you can see, the thickness of the wall is improved and the space in the ventricle is significantly enlarged. So it seems to be working pretty well. Now, there are, of course, questions that you may have. So what about the gene delivery? So gene delivery has been seen to work by giving the gene through adenoviral vectors. And this is the Danon study that we will discuss with Dr. Rossano right now. And it's possible to do it by IV administration. There are issues with immune responses. For instance, if the patient have a pre-existing significant amount of antibodies against this virus, that may happen because they are around. That may decrease the efficacy of the treatment. So that's the need of immunosuppression, one of the reasons for immunosuppression. And how much do they last? So once the therapy is done, how much can it last? So, of course, we don't have data in cardiac diseases because the cardiac field is very new. We have data about other diseases in which gene therapy has started many years ago. So in hemophilia, for instance, over 15 years. In spinal muscular atrophy, over 7 years. In myotubular myopathy, 6 years. So there is hope that actually the duration of the treatment can last for a very long time, maybe lifelong. The other important question that came up from the discussion is, this is a virus, so is this transmissible? Okay, so all the evidence is that there is no transmission once the patient receives the adenoviral vector. However, there is a potential hypothetical risk of seroconversion due to fragment of the virus, that the viral vector. And that's why this can happen to secretion or excretion of the patient. So for this reason, this justifies also the need to have the patient hospitalized when the treatment is given. So just to wrap up everything, and certainly this will prompt more questions, but the goal of the gene therapy is to correct the genetic cause, and which means to prevent when given early, or reverse the condition when the patient already has the condition. The purpose is to do a single one-time dose with a long-lasting response. And we expect that this will improve the heart rhythm, the heart pump, the symptoms, and the quality of life of the patients. The current, where the research is currently, so there is, as we discussed, new trials are in the clinical setting for hypertrophic cardiomyopathy concerning myosin binding protein C3. And there is a hope also in the correction of the mutation now in preclinical animal studies. And with this, I will stop here. Thank you.
That was wonderful and fascinating. And probably a little mind scrambling for some. I know it took me a couple times to kind of learn about this to absorb it. We do have a couple of questions before we head off to hear Dr. Rosano. The first question I'll answer at the end. Just so y'all know, one of our from someplace else is from Brazil. So Brazil's in the house tonight. So welcome, Brazil. Sue has asked the question, how is the viral gene removed? So let's repackage that question a little bit. Sue, I'm going to guess what you were saying here is once you've put a virus into the body, how do we process that virus? And can you explain what an AAV9 and virus and how those terms come together?
So I will explain for what it's my knowledge, and I'm not one of the scientists that are preparing the viruses. But so the knowledge that this particular type of adeno-associated virus, the type 9, is particularly attacking, let's say, the heart muscle. It's a knowledge we have for a long time, I would say. And it has been used, for instance, in the research in the lab. But now it's very successfully used for targeting specifically the heart, because as you can imagine, one of the problems is that you don't want to have off-target, what we call off-target effect. So you don't want this a viral vector containing a particular gene that is very useful for the heart to end up in the liver or end up in another organ. So the expectation is that this is a very specific virus that will target only the heart. And the way technically is done, it's certainly the purpose of another talk. But it would be very interesting to learn also from the companies that are producing these, how their technology is.
Okay, so we have a clarification, then a couple of other follow-up questions. How is the bad gene removed? So I think this is a replacement question. And then there's a follow-up question from somebody else. How do you make the good genes?
It's actually very, very important question. So this is, again, let me remind you that this is the future. So we are not there yet in terms of repairing. So what is called gene editing, repairing the gene. So the way it's done is that instead of putting the normal gene into the viral vector in the package, let's say, what is inserted into the package is the machinery to repair. So that's the concept. There are still concerns and issues that the researchers and the drug companies have to solve in terms of making sure that that's not going to affect other organs. Same problem as we discussed before. But if this is occurring, so the machinery is able to cut the selected piece of the DNA containing the gene that you want to repair, cut it out and replace it with the normal DNA. So that's the basics. And so, of course, this is the ultimate ideal situation. Again, as always happens in medicine, a lot of aspect that needs to be perfected before being safe for human studies.
So I've just gotten a note here from somebody from one of the companies who's watching, and they said, "Oh, how the material is made. That's a great webinar on its own." So I'm going to hold her to that comment. I have written proof. So we'll probably bring you another session on how this is actually derived and what I've been to the plant. I've seen the plant where the gene therapy is made. Do not ask me to explain the entire process. My mind was boggled when I left. But I know very smart people put a lot of very good thought into how this is done. And there's lots of big machines and they get littler machines and it's very sterile and it's like crazy sterile. And I want to make sure good people keep manufacturing this and it doesn't get degraded in any way. So we need really good quality labs for this work. I think that's really important. So I am going to answer a couple of these other questions, or we're going to answer these questions real quick, and then we're going to get on and we will get to all the questions by the end. Phil, we cannot do genetic therapy unless we know the gene. So the question is, what about somebody who doesn't have a known variant? So this is why we're starting with gene testing. Do you have an identifiable gene? Do you have an actionable gene at this time? Or are we going to put it on the list and get there hopefully someday? Do you guys agree that that would be a good way of looking at that?
Absolutely.
Okay. All right. So Phil, sorry, I didn't have a better answer for you or one that fits your situation better. My sister had a gene test, did not identify a mutation. Have some of them not been identified. 60% of people who get genetic testing for HCM will get a no mutation identified result. So only 40% of those with HCM have an identifiable gene at this time, July 2023. And at the time I said it, it was 7/23 and 7/23. So check back regularly to see when new genes are found. There hasn't been any major developments in a few years now, but you never know when something's going to break. Okay. I'm going to pause the rest of your questions. Keep your minds running. If you're asking questions in chat, please bring them over to the Q&A. Not trying to be difficult, but we can actually keep all the Q&As. The chat disappears after the webinar. And this way, all of our questions are in the same spot so we can use them for other purposes later. Dr. Joe Rosano, we're giving you two heavy hitters. We're going to merge the two talks into one. They showed up on the agenda as separate items, but we're just going to kind of talk through all Joe's talk. And then we're going to come back for Q&A. So without further ado, oh, I love the picture. So cute. It's a kiddo.
Great. Thank you. You can see the slides and hear me okay?
You're great.
All right, perfect. And Louisa, that was a wonderful talk, and hopefully this will dovetail nicely. And I would love to attend the webinar on manufacturing for gene therapy. It's a real challenge to make these vectors, especially to try to do this at scale. And so, yeah, it'd be a very interesting webinar. It's a couple of disclosures. And so what I was going to talk to you today was a little bit more about AAV9 therapy for cardiac disease and really focus on what we know about Danon disease, and then also touch on the role of immunosuppression for gene therapy and why we do it and why it's important and what we know about it. And I'll just preface this all to say there's a lot more that we need to know about all of this. So Louisa showed some very nice examples and figures of how gene therapy works, but just to kind of review this briefly again, basically a new gene, in this case, hopefully a normal copy of a gene that there's an abnormal copy of, is packaged into this adenoviral vector, which is then brought inside of the cell, and that normal gene can then inside the nucleus of the cell be copied and have normal proteins be made, and that can in the ideal sense restore health from the disease state. And as Louisa had mentioned, adeno-associated virus, AAV, is a very common vector used for gene therapy, and it's really an interesting story in kind of the history of medicine. So this was first discovered in the 1960s, and AAV, as Louisa had mentioned, by itself does not cause human disease. So this was discovered in laboratories, and they do cause diseases in other animals. But the research into AAV9 was not driven because of the thought that this would be helpful for gene therapy, 50 years later. It was really just to try to understand this virus better. And as much of science was really just trying to understand, well, how does nature work? This looks new, and trying to understand it. And it's just a little bit of a lesson to me in that you never really know what's going to be valuable in the future. And it's a challenging funding medicine, it's a challenging funding science, but it's really important that as a society we continue to do this, because this vector is very important for gene therapy. And had this work not happened over 50 years ago, we'd never be in the place where we are today. And nobody at that time could have envisioned how important this would be to potentially curing many diseases. So where we are today, that this recombinant AAV is really a leading platform for gene therapy, both for these, cardiac conditions that we've talked about with Danon disease and sarcomeric hypertrophic cardiomyopathy, but also for a myriad of other diseases. And gene therapy overall as a field has really, it just exploded, over the last 10 years and even over the last 5 years. There was a lot of interesting work going on in the 1990s and some, pretty significant setbacks with the death of a pediatric patient actually here in Philadelphia. But investigators who really believed in this as a potential therapy were able to pursue through these very dark days. And then, 5-10 years ago, saw some major successes in, certain diseases, which really led where we are today, where there's just an explosion of research and an explosion of interest in that. So this is an article from about a year ago, which just I think was interesting to show the number of current studies that are happening for cancer and genetic disease, as well as for infectious diseases. But if you look at the number of phase 1 studies, so these are early studies, really looking at safety, is this therapy safe? And, is there some signal that it may be helping? Phase 2 is really trying to get a better sense of, does this therapy actually do what it's intended to do? Meaning, is it providing a benefit to patients that have the disease? And these phase 3 trials are really thought to be kind of the pivotal studies where we really learn how effective is this therapy and can we apply it more broadly. But if you look at, for some cancer and genetic, therapies, over 900 current active trials, if you look at clinicaltrials.gov. So, really an explosion in interest and excitement about gene therapy. And I'm not sure how well you can read this figure, but it's a pie chart that just shows the different types of vectors that are used, including non-viral vectors, but adenovirus, makes up an AAV make up a significant number of vectors that are used. And the reason that AAVs are used is because they have fairly high tropism for different organs, meaning that they have an affinity to these organs and will bind and can be deliver gene therapy to these areas. So if we look at AAV9, if we look at AAV9, it has quite a bit of tissue selectivity for things like heart, skeletal muscles, retina, and the central nervous system. And so it has been used for things like spinal muscular atrophy, which is a central nervous system disorder, and has been used for things like Duchenne muscular dystrophy, which is both a skeletal myopathy, but the leading cause of morbidity and mortality in Duchenne muscular dystrophy today is actually a cardiac issues. And you may have seen this just recently in the headlines, but from the end of June of this year, the FDA approved the first gene therapy for treatment for patients with Duchenne muscular dystrophy. So this was, very exciting for the field. I will say it's still a bit unknown of how effective this therapy is going to be, both in terms of the skeletal muscle, so the arms and legs, functioning of boys with Duchenne muscular dystrophy, but also for the heart. The heart generally with Duchenne muscular dystrophy becomes affected later on in life, sometimes severe cardiomyopathies don't develop into the second or even a third decade of life. So it is really unknown at this point, does this therapy, which is an AAV9 therapy for Duchenne muscular dystrophy, will this have a beneficial effect on the heart if we're treating patients pretty early in life, at age 4 and 5, before we would see any evidence of at least a moderate or severe cardiomyopathy. But certainly very exciting for the field to have this approved therapy. So what do we know about Danon disease? So Danon disease is a monogenetic X-linked disorder that is autosomal dominant. And what that means is that the gene is carried on the X chromosome. So men would have an X and boys an X and a Y chromosome, and women would have 2 X chromosomes. So for boys and young men, if they have an abnormal copy of this, with an X, only one X chromosome that has the abnormal gene, they can become affected pretty early in life. The disease which causes Danon disease is LAMP2, which is the lysosomal associated membrane protein, which is a very important gene which is involved with autophagy. And what autophagy is, it's basically the cell's way of getting rid of old proteins and other cellular byproducts that are no longer functional. And what autophagy means is just literally means self-eating. So it's the process by which the cell will, quote unquote, eat these senescent proteins and get rid of them. And when that is impaired, there will be this proliferation of lysosomes and accumulation of byproducts within the cell, which will lead to disease. And I'll show some pictures of what that looks like in a minute. And what this leads to is a severe cardiomyopathy. Males present very early in life and have a high mortality in the second or third decade in life. I'll show some figures in a minute, but really for boys and young men with this condition, it is unlikely that they would survive past their early to mid-20s without a heart transplantation. Females, which people that have 2 X chromosomes have one normal copy, one abnormal copy. They can also develop disease, but tend to develop it later in life, and their course is also more variable, meaning that there are some severely affected individuals at early in life, and others that are less affected even at older ages. And what is striking about Danon disease is just the severe and massive hypertrophy that develops, especially as people age. It may seem as a mild or moderate form of hypertrophic cardiomyopathy, where patients are in the 8 to 10 to 11 years of age, and there can just be a rapid progression of the thickness of the heart, as well as symptoms of arrhythmias and heart failure. And if we look at, this is just a cross section of a heart for somebody with Danon disease that shows just a massive hypertrophy. And if we take a section of that and look under the microscope, these white areas here are these abnormal vacuoles. So these accumulation of abnormal cellular proteins and other byproducts that really impairs normal functioning, and you have very abnormal myocyte structure. So the normal, what's called the sarcomere, where the way the cell looks at the heart muscle cells for as it contracts, have these regular kind of uniform appearance to them. That becomes disruptive. And what's notable clinically about Danon disease is the typical medications and strategies that we would have for people that have sort of typical hypertrophic cardiomyopathy or hypertrophic cardiomyopathy caused by sarcomeric proteins. The medications that are available do not change the natural history of the disease, meaning the disease progresses in spite of these medications. They may be somewhat helpful for symptoms, but the disease just progresses on and really leads to severe arrhythmias and heart failure very early in life. And this just kind of shows, dramatic example of what a severe disorder this is, especially for male patients. So this blue, I just ask you to focus on this blue line here and this orange line. So this blue line are male patients that are diagnosed with Danon disease. And this is what's called a Kaplan-Meier curve. So up here, at 1 is 100% survival. So you would like a disease where this just was flat across, everybody survived over time and nobody died. And as this curve goes down vertically, these are patients that are no longer alive. And you can see that males with Danon disease, the vast majority of them, don't survive or need a heart transplant by the time they're in the earliest to mid-20s. So really a very, very severe cardiomyopathy. And what we see from women is that also can be a very severe disease among women too. I will also say we know far less about women with Danon disease than we do about boys and men, although it is a rare condition. So there's certainly a lot we don't know about boys and men as well. But women have a more variable prognosis, tends to be later in life, but still pretty significant number of women that either succumb to the illness or need a heart transplant by the time they're in their 30s, 40s, and beyond. So we're really excited to be in a place where we have now completed a phase 1 gene therapy study for Danon disease. This is really a fascinating story. I'd be happy to answer any questions about the development of this. I think Eric Adler participated in the webinar for the Hypertrophic Cardiomyopathy Association about a year or so ago. And he is a cardiologist and scientist that really developed this therapy. He's been studying gene therapy for Danon disease for essentially his whole career, and developed a gene therapy product and tested it in a mouse model of Danon disease and had excellent results. And then was able to partner with Rocket Therapeutics, and a lot of hard work got to the place where we were able to perform a phase 1 study of gene therapy for these patients. So this cohort consisted of both adults and adolescents, which were 15 years and older. And we were able to enroll 5 patients in this cohort, as well as a pediatric cohort that were age 8 to 14 years old. And we enrolled 2 patients. Those patients were at my hospital, at the Children's Hospital of Philadelphia. And there was a high dose group that received 1.1 times 10 to the 14 genocopies per kilogram. And 2 subjects, adult subjects, received this. And then the rest of the subjects received a lower dose of 6.7 times 10 to the 13th. And the doses are important when thinking about both the effectiveness of the therapy, but also in terms of the risk of important side effects. So I just want to spend a minute to talk about potential side effects. And, gene therapy is still in many ways a field in its infancy, and we're learning a lot about it. But there have been very important serious side effects, including deaths in some studies and in some patients that have received gene therapy in clinical use from AAV9 vectors. Some of these deaths and adverse events have been from what is referred to as this complement-mediated phenomenon. So a complement is part of our innate immune system, and our immune system can get revved up after gene therapy and can cause this condition called thrombotic microangiopathy, or TMA. And what can happen is that blood clots then start forming in our blood vessels. And in small blood vessels, they can have a lot of these blood clots. In some patients, it's led to renal failure because of this injury to their kidneys. Can be reversible, but it can be serious and can lead to death. There has been liver toxicity in some patients that have received gene therapy. What is a potential concern, especially in the cardiac realm, is myocarditis, or an infection in the heart. And at least a couple of deaths from gene therapy for a Duchenne muscular dystrophy were thought to be cardiac related, and potentially this inflammation in the heart. As well, skeletal myopathy has been seen, meaning weakness in the arms and legs. That may be in part not necessarily from the gene therapy itself, but from the medicines that are used to try to blunt the immune system, like steroids. A Danon disease, I should have stated, it does not exclusively affect the heart, although the heart is the organ which leads to the major morbidity and mortality. But patients can also have a skeletal myopathy. It's usually, relatively mild, but they do have and can have weaknesses, in their arms and legs and their so-called skeletal muscles. It can also have some CNS abnormalities, and children can have some developmental delays. This is variable, and it can be quite minor. And there's also retinal disease. So even though the heart is the primary organ, it is a diffuse and systemic disorder. And so because many of these side effects are thought to occur because of the body's response to the gene therapy product, to the viral vector, immunosuppression is used to decrease the risk of developing these. And what we've seen over time, both through the Danon study as well as through other studies with AAV9, that the strategy and approach to immunosuppression has changed. And we hope and we think that we're on to a good regimen, but it's something that's really under still under evolution as we learn more about the potential side effects. So what do we see specifically in the Danon study? Well, one patient did develop this thrombotic microangiopathy. This was an adult patient who was one of the subjects that received the higher dose. Because of this, now only one patient, so it's hard to know how common this side effect may be, but the remainder of the subjects, and then what is planned now for the phase 2 study, is actually on the lower dose of the gene therapy, which seems to be as effective and may be associated with lower side effects. As well as the immunomodulatory regimen is different now than it was when this person received that. Fortunately, this patient recovered, but did need dialysis for a short period of time. As well, some of the adult patients developed a significant skeletal myopathy. This is probably related to the steroids that they were on and being on higher doses of steroids for a longer period. So that was also changed over the course of the study. And I can say that the 2 pediatric patients that we dosed really did not develop any, they really didn't have any significant adverse events at all, including, no significant skeletal myopathy. And so, again, we didn't really see any serious adverse events in patients that received the lower dose. And just one slide about this immunomodulatory regimen, and I'm happy to answer any questions if people have about the medicines and why we use them. But again, it's a primary aim is to prevent this immune-mediated complications of gene therapy, including this thrombotic microangiopathy. It may help to prevent this concern about myocarditis, but I think there really is still a lot we need to learn about what caused that in those patients that receive that, and what we can do, to prevent it. At least in some subjects, it seemed to be reversible with no lasting damage. But again, still a lot more to learn. And the current regimen for patients in the Danon study are to receive steroids around the time of receiving the gene therapy, and then those are, relatively quickly tapered. Rituximab, which is a medicine. Actually, all of these medicines are medicines that we oftentimes use in our.
Uh-oh. We've had a temporary glitch. I'm going to pop in here for a second while Joe comes back. Here we go. We're back.
Okay.
It
Sorry about that.
happens. That's okay. Technology just keeps us on our toes. Make sure we're listening.
All
I'll hand
right.
it back to you.
Okay, thanks. Well, my dog has joined us, and sometimes she turns off my computer, but she seems to be listening intently now. So hopefully that will stay that way. But I was just saying that the regimen we use is a regimen that are medicines that we commonly use in our heart transplant patient, and rituximab works on a certain type of our B lymphocytes, which help to make antibodies. And sirolimus is another immunosuppressive medication, which we often use in heart transplantation. And these are tapered and removed after the first couple months, after we think at least the risk of any of these immune-mediated issues are behind us. And just some early findings, and I will say these are early, but it's very encouraging. I will admit, I'm a part of the study, so I probably have a bit of a biased opinion, but I've been very impressed with these early findings. So we do see gene expression, which is important. I mean, ultimately, we're trying to replace a non-functioning gene. And through biopsy specimens, we do see that up to 36 months of some of the adults have been followed that long or even longer now. We do see gene expression, as well as we've seen, some improvements in functional capacity as measured by New York Heart Association class, which is a common sort of way that we grade heart failure. And there's been some encouraging findings with the amount of hypertrophy, as well as with biomarkers, things like natriuretic peptides, which you may be familiar with, is an important biomarker in cardiovascular diseases, and generally the lower the better. And we have seen either a stabilization or reduction in most of the subjects. And so we're very excited and encouraged by these phase 1 results, and a phase 2 trial should hopefully be enrolling very soon. And this is just a slide from that I presented regarding our phase 1 results at the ASGCT meeting a few months ago, which just looks at the natriuretic peptide levels of subjects in the study from the left. And you can see that really went down, or if they were low, they stayed low, which is at least in a very small number of a small number of patients where we've prospectively been able to follow their natriuretic peptide levels as part of a natural history study. They just go up over time. So again, very small numbers, preliminary information, but encouraging. And we will certainly learn more with the phase 2 study. Also, just want to say that there are important similarities as well as important difference between, what is the most common form of hypertrophic cardiomyopathy, meaning sarcomeric hypertrophic cardiomyopathy. Whether you consider Danon disease a form of hypertrophic cardiomyopathy, is debatable. They both have very thick hearts, but the etiologies of the diseases are very different. They are both monogenic disorders, and they lead to thickened hearts. And the major morbidity in these conditions are driven by cardiac disease. But they're very important differences, which I think do have important implications for how gene therapy may be studied and ultimately may be used. Sarcomeric HCM, for many patients, there is effective medical and surgical therapies. Now, that's obviously not true for every patient, and that they do not respond to medicines, and they're not candidates for surgical therapies. But that doesn't exist in Danon disease. There are no effective medicines and there are no effective surgeries, as well as, it's quite a different natural history. The natural history for boys and young men with Danon disease is different from women, and it is very different from sarcomeric HCM, where there's a huge spectrum of disease. And sarcomeric HCM is also much more common, but there are people with mild disease who may fare well for long periods on no medicine or little medicine, and then there's very, very severe disease that can affect people very early in life, even in their first year of life. And so that it is important when we think about what's the best population to study it. If it does look effective, what's the right population to give it? When is the right time to give these medicine? Is it better to give it early before there's severe disease? Is it better to wait until there's some evidence, or if we know who's going to respond well to medicines and who's not going to respond well to medicine? So, again, there's a lot that we still, need to learn about this condition and about this therapy. So, in summary, gene therapy with AAV9, I think is incredibly promising therapy, not just for Danon disease and sarcomeric HCM, but for many cardiovascular disorders, especially if they're caused by a single gene that is responsible for it. And the early experience with Danon disease, again, I'm probably biased from being a part of the study, but I do think it's been a very favorable experience. And as Luisa, had mentioned, there is, interest in expanding this into more common cardiomyopathies, and the MYBPC3, mutations is, there are active studies being planned, which will hopefully be enrolling soon. And, if that does prove to be effective for, that condition, for those mutations, that's, a huge breakthrough for the field, as, as obviously is a very common mutation, a common a cause of hypertrophic cardiomyopathy. But key to all of this is that we need to do the ongoing work, and we are doing the ongoing work to understand the safety of gene therapy and the efficacy. And when is it helpful? Who is it helpful for? And what is the best timing to use these therapies? So with that, I will stop, and I'm happy to answer any questions.
Oh, they've got questions. Luisa, come on back, because we got questions for the whole crew here. Okay. So it's going to kind of be potpourri because they pop around a little bit, but I want to just get some general agreement on some topics because they've popped up in a number of the questions. If somebody were to participate in the clinical trial with an AAV9 delivery system, number one, will they have any potential to pass the genetic mutation on to offspring? Yes, correct. Who wants to comment? Does the AAV9 alter the genetic code in the reproductive system so that you're not passing it on? No.
No, as I mentioned, and it was one in the slides. No, that's an important thing to know, because the heart will be repaired, but not the whole body. So, and not the reproductive system.
But the abnormal copy of the gene could be passed on,
Right?
Correct.
not the corrected copy, not the normally functioning gene that was delivered by gene therapy would not be passed on.
Correct. Okay. So we discussed in the last question break that only those with a known mutation can participate in this particular trial that we're talking about today for myosin binding protein C, but it's going to be critical that you know your genetic mutation, which kind of ties into the first question I was asked, which is a little above my pay grade, but I'm going to try to address this. Will the HCMA legislature, we don't have a legislature, improve, I think you mean my legislative committee, the availability of genetic testing without insurance standing in the way? So in 2005, when the first genetic test became clinically available through a clinically available laboratory, at that point it was Correligen and Laboratory for Molecular Medicine. The price was about $8,000 for 3 genes. We have helped, and we have walked side by side with genetic testing companies to help develop bigger panels. We've brought down the price, and the average individual can now get a panel that has at least 70 genes, if not more, for about $300. And it is covered by most insurance. Medicare is a little funny about it, but the out-of-pocket cost for genetic testing today has come down, not just because of the HCMA and us yelling at people and begging them to bring it down. We've modernized, and we can use computers to do a lot of this analysis now. So the price has come down, availability has gone up. It is known to be very effective. And And insurance companies will always be annoying. Sorry. Some of them will kick out claims that are very legitimate, and you can put in an appeal, and you can go to a third level appeal if you are the index patient in the family. We can try to help you with that fight, but we can't make every insurance company do what we want. If so, then I will have a lot of changes in store. My guests, do you agree with what I've just said? Okay. So anonymous attendee, I have answered your question. There we go. And we've answered that one. If somebody, this is going to be a lot of ifs and hypothesis. If somebody had genetic therapy and it met the destination, it gets to the heart, and it helps the heart to function more normally, would that person then potentially be able to discontinue medical therapy? In this hypothetical world that doesn't exist today, because we're only in a phase 1B clinical trial, hypothetically, could that happen? I'm going to start pointing at people if you don't talk.
Yeah, I'll try to answer that. I mean, the hope is for a cure. I think it's too early to know that for Danon disease, and we haven't, the NYBPC3 studies haven't started yet. There's probably, for most people of what we've seen so far, there's not equivalent gene expression to somebody that didn't have disease, but we don't know how much gene expression does somebody need to have a significant improvement. I mean, we have some idea based on women with Danon disease, but it's we just don't know. And so I think it's too early to know, are we going to cure disease, or hopefully if it's not a cure, but really improve what would be the natural history, meaning that people will live longer, they'll feel better, with medicines or without.
Good answer. The next one's going to get a little bit more complicated. Is this therapy easily transferable to variants in other genes like myosin heavy chain? Luisa, your turn.
Well, I mentioned this. So
Yeah.
the problem is that for the replacement therapy, which is what we have now, you need to have a small gene that can be put in the package. And you need to have a situation in which there is a lack of amount of the normal protein, so that the gene that we package and give to the patient can start producing more of the normal protein, let's say. The myosin heavy chain, so MYH7, is the case which I mentioned, in which the attempt is actually to repair, so to do a precise repair of the mutation that exists. And the reason is that this is a really large gene, and it's very difficult, if not impossible, for the current technology, which may change in the future, to package it and deliver. The other problem, as I mentioned before, is that in this case, there is not so frequently a problem of amount of protein that is missing, is the defective protein that is produced because of a mutation. And so the only way actually to address that question is to repair the mutation, basically. So this is the object of intense research now. When it will be available, we don't know. There are still a number of issues and safety issues and precision repair that need to be addressed.
Okay. The people on this call cannot exactly answer this question, but Susan, I will address it for you. Why is there an age cap of 65? I'm young at heart, because there has to be endpoints for certain types of trials, and they've opted at this moment to cap it at 65 probably has a lot to do with comorbidities as we age, and they want to keep it as clean as possible. I know your heart is beautiful, and thank you for your willingness to even consider using it for this trial, but hold tight. And if they change the number, then maybe you can join. Okay. So if anybody has anything to say on that, I will add it. What are the target completion dates for each phase of the study? It doesn't work that way. We have a start date and numbers and safety signals that need to be met in any clinical trial. So the first patient will be dosed as soon as August, which is tomorrow, but it's not happening tomorrow. But it could happen by the end of August. You will not probably hear about it right away because we're going to want to give it a little time to see how the person does. And then there'll be an announcement when it is appropriate that it has happened. They will wait, I believe, 30 days between dosing different patients in the first cohort. And there will be 3 patients in the first cohort. There'll be 3 patients in the second and the third and the fourth, I believe. I think it's 3 or 4 cohorts of 3. And they will evaluate each step of the way. So we're probably not going to be completed for probably over a year, because you got to give 30 days between each dose or thereabouts. And unless things start to move really well or things go really poorly, and then everything will change. So you'll hear updates as the sponsor is willing to put them out. It is a publicly traded company, so we have to, there's all bunch of regulatory language involved here as well. So when they're ready to announce findings, they will let us know, and we will let you know when those announcements are going to happen. So we will keep you in touch with the latest as quickly as we possibly can bring it to you. Any comments on that? That's kind of how they work. It's going to be a while. It's not happening literally tomorrow, so please don't call me in 3 days and ask what happened. What is the best way to keep up to date on developments? Keep up with the HCMA, our newsletter, our website, our social media. When there's news, we will be letting you know. Positive, negative, we're just going to be transparent. Let you know what we know when we know it. How does one find out? Oh, okay. How does one find out if they have an AAV9 gene? Does Medicare cover such costs? Louisa, you want to help me out here? It doesn't work that way. It's pretty much
Doesn't work that way.
the starting question.
So the question, if I understand correctly, is how do I know that I have already antibodies, for instance, against the AAV9? So in fact, in the protocol, one usually in the very, very initial phases of one of these studies, the very first thing is to study how the population of patients has in terms of amount of these antibodies, if there is any particular amount that can be a problem for the therapy. So this is part actually of the trial. And what I probably should remind our patient is that we are talking trials, so we are talking still studies. We really hope that in a few years this will become a standard therapy, but we are not there. So we still are in a study phase. So that's have to be kept in mind.
Okay. Joe, did you want to add anything to that?
I was just going to add that, I think now for most, or certainly all studies that I'm aware of their protocol, it's an exclusion criteria. So if you have this antibody at a certain level, you'd be excluded. But there's interest and there's, experimental studies looking at how you could, blunt that response. So, hopefully if this, these therapies become approved in the future and somebody does, have a neutralizing antibody, they could be sort of preconditioned before, ahead of time so that the therapy, could still be made available to them and still be a benefit.
Fantastic. We have a question. Why do you have to have a defibrillator to participate in the study? And they have clinical trials for those who want therapy but don't have defibrillators. My gene test results meet the requirement, but I do not have a defibrillator. I know as a safety signal for the first in humans, They want those with defibrillators. That may or may not stand through all of the cohorts. I know cohort 1, definitely they want a defibrillator because they don't know what the heart is going to do. They want to make sure if it's going to have an arrhythmic response, which is highly unlikely, that there's a defibrillator on board. So stay tuned. Those criteria may change in the later cohorts or in a study that may be brought to you by a different sponsor. There may not be a requirement on that one. Any other comments on that before I get on to the second question? Okay.
The
Second.
more we will know, the less requirement we will have.
Exactly. Once we know where you're safe. So I think something I said was misinterpreted, so I'm going to be clear on this. The question is, you said gene therapy is a type of machine. Is the machine known as nanotechnology? It is not a machine. It is a system that creates the very tiny molecules that go into tiny, tiny little ampules that are the protein. And there is machining or equipment that is responsible to bring these proteins into the world through lots of technology that I don't understand completely, but I saw the equipment itself. It goes from very, very big things to very, very small things. And they're silver and they're big and they're shiny and they're brand new. And they haven't really been used much. And they have lots of refrigeration there too. So it is not an actual machine. So there's no machine in your heart. There's nothing there's nothing mechanical being added to your DNA. It is a protein that is created in these systems and in these vats. Guys, did I explain that one well? Okay. My son is 8 and has severe HCM. He also had a septal myectomy due to a recent cardiac arrest. Okay, anonymous. Will gene therapy help and serve HCM or only those with Danon's? So, and I hope we can see clinical trials soon. So in Danon's, we were able to do clinical trials in young males because the disease is deadly if we don't do something. In HCM, we do not typically have the same level of catastrophe early, and we have more time. So there's probably not going to be a lot of tolerance for clinical trials in young people until we know how it works in road-ups. Would you guys agree, disagree? Any additional comments there?
Yeah, I agree. And, the Dana protocol, even though we know, boys are affected, it starts in the older age group, and then wanted to make sure it was, safe and before we went down, to the younger ages. And that, but we won't do that in the phase 2. At least that's not the, plan, because, we think we think we've learned enough about the, lower, younger ages. But to the person that asked the question, I hope your son is doing well. And but I do hope these therapies, one, we do study ultimately in children, and that they are made available to children as we learn more about them. So it's probably still a bit of a ways off for sarcomeric HCM for delivering these to children, but hopefully it is on the horizon.
I would agree. Okay, so this one's going to take a little bit. Is this the same type of machining that was used to create the COVID vaccine? Because of the same dangers Dr. Joseph talked about with some of the outcomes from COVID vaccine, blood clots, myocarditis, etc. Who wants to take the similarities and differences between a COVID vaccine? I'm going to pick on Joe because he's not on mute.
Well, it's there, I think there are important differences. It's not a vaccine, the intention is to, replace a, defective, gene in the body, not to make, antibodies against a, a virus. And I'm not a vaccine expert, but I don't believe the, side effects that were seen from COVID were really thought to be complement mediated phenomenon, but Louisa or others maybe could correct me. So anyways, I think, important differences. I don't think we know, we still have a lot to learn about the potential side effects and the ways to mitigate those side effects for gene therapy, especially for, cardiac conditions and, muscular dystrophies. I think that is a very important part of these studies, is to understand the safety, where the risks are, and how to mitigate the risks. Which is not that that's not important in studying vaccines, but it's different. The COVID vaccine was given to millions of people around the world, and so we learned quite a bit about it. Generally very safe or very safe. And we just don't have those numbers, for AAV9 for cardiovascular diseases, and so we just have a lot to learn about it.
So one of the side effects that has been spoken about is myocarditis, which is a general issue that can happen from a lot of different reasons. Can one of you discuss why the heart might inflame with a virus, any virus? Louise, I'm calling you off of mute because poor Joe's like, please don't make me do this again.
So the reactive myocarditis can happen in a number, as you said, so in a number of situations. It's actually interesting. So we don't know exactly the mechanism, and so cytokines, inflammatory response that maybe justify myocarditis, by the way, can be the cause of arrhythmia, and that's why, for instance, the requirement for safety reason of the ICD in the trials, until we know what can be the association, why some patient may develop myocarditis rather than other. And usually it's rare, but it may happen, yeah. But as Joe Rossano said, so we really don't know many of the mechanisms. We are still in a learning phase.
Absolutely. So somebody was tested for a variant of uncertain significance. How often should they be retesting or reevaluating that variant? Annually with their physician, more frequently?
That's a question I get every time. So some of the big laboratories actually review constantly the results, because sometimes variants that were considered of uncertain significance can change the significance, can become disease causing, or can be clearly benign. So it's usually up to the laboratory to update regularly and to let the physician know, and therefore also the patient. It's the other question is that the more we learn, so the more expanded the panels are, as you were mentioning, Lisa. So in the past we have 3 genes, so now the panels are hundreds of genes. So something may change. So to contact the cardiovascular geneticist or the geneticist every couple of years can make sense.
I was on a group that was evaluating how we should manage variants of uncertain significance. And it was, I got to tell you, it was 3 years. We met a few times a year. We kept banging our head against walls as to the legal, the clinical. When does a relationship start? When does a relationship end? How long is this relationship? Who is the relationship between the doctor and the patient and the lab and the doctor? So we asked a lot of really interesting questions. It's been published, and we need to figure this out because there's a lot of variants of uncertain significance, and some of that information is going to never work out to be anything meaningful. And in other cases, it should work out to be something meaningful. So behind the scenes here at the HCMA, we get involved in a lot of things, and that's one I got dragged into. It was a really interesting conversation that didn't really have a lot of concrete solutions, just a lot of things we all need to work on. Does it matter where the mutation is in myosin binding protein C for this particular study? This is an excellent question, Alice. 2 points to you. I'm going to start with Louisa, then I'm going to go to Joe on this one. So what are the different mutations that could possibly be there?
So in the case of myosin heavy chain, you can have both what is called loss of function. So the gene is not produced, and so there is lack of protein, or there can be what is called missense mutation. So just one single base, the code is wrong, let's say. The code of the gene is wrong, the protein is produced, but the code of the protein is also wrong at that point. And so that's why in that case, the strategy has to be slightly different compared to the trials we have available now. And that's the requirement of more studies and the ability to really repair that particular single mutation.
So this questionnaire was about myosin binding protein C. So I just want to be clear on
sorry.
that one. Yeah. So does it matter whether with this particular, with Tenaya's therapeutic option for the clinical trial, does it matter if it's a missense, a deletion? Does it matter what kind of mutation we're dealing with here?
I have the protocol here, but I don't think it has to be a loss of function. So it cannot be just a missense because it's again, it's a replacement type of strategy. But the particular location, I don't think that that's influence.
The location isn't, the type of mutation is important.
Right.
And if you think you might be interested in learning more, this would be a good opportunity to kind of give a pitch to speak to a clinical trial coordinator. They can help assess your clinical records and determine whether or not further conversations are warranted. And you just start by saying, yeah, I'd like to learn more. And you take it as far as you want to take it. And you may learn enough and say, yeah, I'm not interested, or wow, this is getting more interesting as I dig in deeper. I think the most important thing about this trial, maybe more so than other trials we've dealt with in HCM, is the participant really has to understand what they're doing, understand the science, has to make sense to them, both in their mind and in their soul, and they can go ahead and try it if they qualify. Joe, any other comments on that?
No, I think that was, well described. You know, they, Luis had a very nice figure. I don't know if that can go out to, I guess it will be, this talk is recorded, but you know, when, you know, there's a loss of function and that's the real, you know, where gene therapy, you know, currently is, you know, I think probably most effective is when, you know, there's not a functioning gene and you can replace it with a functioning one, you know, you know, very exciting things, but still, I think, you know, preliminary or early, you know, she described as, you know, if you're trying to edit a gene, that's, you know, making a protein, that's, you know, still a bit in the future, probably.
So I have a question coming from in-house here at the HCMA. How does dosing work with gene therapy? What amount of the gene protein goes into the vector? What do we know about dose size?
Great question. So, you know, higher doses, you know, probably deliver more. Well, they do, you know, you're giving more vector, but more vector can also be associated with a greater degree of side effects. And so you really want to try to find out, you know, what is that sweet spot where you can deliver enough to have, you know, affect the disease, but not have toxicity. Another, you know, important part of that, and this really gets down to the manufacturing, and I wasn't getting that. I'd love to see the manufacturing webinar, but there can be quite a variability of how many empty vectors there are, and their manufacturing, you know, challenges, and so it really does make a difference of how it's manufactured, and the more copies, you know, you can have, so not having empty vectors, which basically can have side effects but aren't going to deliver, you know, have no benefit. And so if you can have that number be very low, and I think it can be quite variable depending on the you know, the manufacturing process. So that's a way to sort of increase the effective gene delivery with the same risk, because it's the same, you know, number of, you know, viral vectors, and kind of my simplistic way of understanding it.
I think that's a good and simplistic, yet not simplistic way. So the questions are going to get a little bit tougher as the night goes on here. Somebody didn't understand one of our answers. And I think there's a core here, Olga, that you have to have a known genetic mutation in this particular area. You can't just give the therapy and hope it covers something else that we don't know what it is. So her question is, what if you have no identified gene? Would the gene therapy work by replacing the entire gene? But we don't know what gene is causing your heart to malfunction. And it may not be a single genetic mutation. There's this concept of polygenetics that maybe 3 or 4 genes are slightly abnormal, but when you put them all together, they make HCM just like myosin binding protein C. So we don't know what those causes in you might be at this time. And until we know, we can't target a therapy there. Guys, did I explain that one well?
Perfect.
Okay.
Yeah.
Gotta
There
keep
are
checking in with the
some- I
team.
mean, there are some people looking, I don't, to my knowledge, not in hypertrophic cardiomyopathy, but for other things like heart failure, to have gene therapy products that are a little more generic that can target things like calcium homeostasis and other things that are deranged in heart failure that could be applicable to a wider range. But, this for hypertrophic cardiomyopathy is really, precision medicine. It's really trying to target this gene is abnormal. Let's replace this gene, which, I think in a lot of ways, what's exciting about it is that it's really targeting what the problem is. And, we've not had that for, most of what we do to treat cardiovascular disorders.
I think some of the most intriguing questions are coming at the end here. What if you have the gene, but no malfunction yet? Can you do the therapy before having an issue?
Great question.
Fantastic question. I will start with the general, maybe someday, but not in a clinical trial. And Luisa, why don't you talk about that a little bit more?
So the point is that we are born with the gene in a certain way. So the gene is there, either normal or defective, since we were born. But it's very right that actually the problem, the disease, the hypertrophic cardiomyopathy can come later. And in many patients actually doesn't really interfere with a normal life. So maybe this patient don't need a gene therapy. So, but the other important point is that certainly the idea is that, and Joe Rossano can correct me, but that eventually, we don't know when, but eventually the sooner the better, because the sooner you prevent any malfunction of the heart, the better it is. So it is possible that in many years, this type of therapies will be given when the patients are very young. So to let the heart remain normal as when they are young.
So I'm going to take 2 questions and I'm going to put them together because it kind of is on the opposite side of, can I do it before the disease presents? We have questions of, well, what if it's really presented? What if you have scarring in the heart? What if you have atrial fibrillation? What if you're already in heart failure? Is it too late to treat? Or do we not know yet? We're never going to get scar tissue to turn into normal functioning myocardium again. It's scar. But is there a possibility that this could be beneficial to somebody with a more advanced? I wouldn't say up to the point of transplant. I'd say we probably need to be a bit before that. So what's enough, but not too much that we might be able to turn back time a little? Does anybody know the answer? I've been looking for it for a while now. I haven't found the answer.
No, I don't. But hopefully we'll learn more over the years to come to get a sense of really what is that sweet spot. I think there probably is a point where it's too late and somebody's not going to receive benefit. I think that's certainly the case in Danon disease, where there's these hearts become so abnormal that even with a functional protein, it's there's a point, and I'm sure with sarcomeric HCM too, that just the damage, there's been too much damage done that it can't be reversed. So you really want to find that time of when you can intervene and have the most benefit But even if you can't have the most benefit, there may be some benefit, and even people that have very advanced disease. And that's what we really, these studies and then also just, assuming it does get approved down the road of sort of clinical practice when, it's made, available, to broader populations. I think that's oftentimes when we really learn the most about how effective a therapy is, when you look beyond the very carefully controlled clinical trial and in the real world, how does this affect people?
Couple other questions. We're wrapping up. Everybody post your last questions because we've taken enough time tonight from these lovely people and we're going to let them have the rest of their night soon. So in the event, again, back into hypothetical world here, the trial is successful, things are looking positive. Do we suspect that the sponsor might increase the age that people could get this therapy once it is a proven piece?
I would say so. Yeah.
I would agree. I think for the purposes of the trial, as you age beyond 65, I know I've got some really fit people out there, but you get a lot of other problems that happen and you want to try to eliminate all the other stuff so it doesn't confuse the science. So they're trying to keep it pure for the beginning of the trial and it may expand over time. This happens in all different types of trials, not just these gene therapy trials. Okay. Are genetic test results consistent lab to lab? How often should you retest? So
once you have a result that is consistent with a pathogenic or likely pathogenic variant of a gene that is causing the disease, there is no actually need to be retested for what we know now. That's a definitive diagnosis. When instead, when you have a result that is uncertain, the variant of uncertain significance, for instance, in that case, the problem could be to be retested.
okay. I'm just going to put another little comment on there. We've been doing clinical genetic testing in the HCM space since 2005. I remember when it was really hot and cool and new and we were going to do this thing and it was going to have all the answers. Didn't quite have all of the answers, but it gave us a lot of answers. And a couple of companies have come and gone. If your testing was done by a company who may not be in business anymore or may have been bought out by somebody and it's turned over a lot, it's been a long time since those tests were out there. You may want to check in with one of the companies who are currently offering genetic testing. That way, if you had a VUS, they're going to be monitoring your VUS now. But Corelligen sold to LabCorp. I don't even know where the legacy files are for their. Familion went out of business. They were bought by Transgenomic and then they disbanded. So nobody really knows where that data is. So, maybe you want to kind of revisit the current companies that are offering and retest. You may find some new information. That's just kind of from the practical girl from Jersey, not the geneticist. It's just kind of hard to find those old results. Diane has 2 questions. Even just guess, when do you think gene therapy will happen? Well, it's happening. Gene therapies are happening all over the place now. We have all kinds of, this is Lisa's transplant drug reminder, because even 6.5 years out, I forget. It's 8:30. Do you know where your drugs are? Yes, they're home. Anybody old enough to remember that? Thank you. When will it happen? So gene therapies are happening now, and they will continue to evolve, and we will continue to do more in gene therapies in a lot of different spaces. When will this particular trial launch? Imminently. And we should have some data in the next 6 months to a year as to what looks like we're trending. It will not probably be published for well over a year, but we will get that information to you as soon as it is possible. Your second question, will surgery and gene therapy help? For some people, yes. They may have a myectomy earlier in life, and they may then benefit from gene therapy. Having a septal reduction therapy does not disqualify you from a future therapy. We couldn't say that. But for this particular trial, for the early myosin binding protein C trials, they want the non-obstructed. I think probably again for clarity and cleanness and not messing with obstruction. We just want to know what it does to the heart muscle and whether or not we can hit the destination and it does anything positive. Once you receive the therapy and the later time genetically tested, well, oh, that's a really good question, Fritz. If you have gene therapy, will you still show up with a gene mutation?
Well, yeah, because your DNA still will contain, for instance, in the blood, the variant. Yeah.
But your heart has received the therapy to replace the missing proteins.
Right.
Okay. Fritz, that was an excellent question. And I'm going to let that be the last question of the evening. I am going to drop in the chat box right now a survey for you to take. And don't worry, my faculty is like, what do I have to do? No, you don't have to do anything. Copy, paste. This is a survey. This survey will give us a little bit of feedback on who watched the whole thing all the way through and who took the time to complete the survey after. And it will also give you an opportunity to say, yes, I'm interested in learning more. And you can give us consent to share your contact information with the coordinator for this particular study from Tenaya, and they will be getting in touch with you. So if you want to learn more, you will have to answer some questions properly. You have to have an active known mutation in myosin binding protein C, and you will have to provide that information and your clinical information to the coordinator, who will then assess whether or not you can take the next step to learn more about this particular trial at this particular time. If they say no, not now, that may not mean not forever. So we're looking for very specific patients for these first 9 people that are going to be dosed with a first in human gene therapy for myosin binding protein C. So our job is to help you learn about it, to answer questions, and to help connect you with those who are doing the research so that we can move this research further faster. So Dr. Rosano, Dr. Mastroni, thank you so much for your time tonight. It was truly enlightening to our population. And I've gotten some great feedback here, and a lot of thank yous are popping through the chat. For those of you who aren't grabbing the SurveyMonkey link through chat, you are also going to get a copy in an email, and you will be able to visit this presentation again on YouTube or Facebook and keep the conversation going. And we will be bringing you another version or another chapter in the genetic therapy saga. And we will be talking about the manufacturing process for the protein because we decided that tonight. And I have text proof of that too. So it's going to be a thing. Guys, thank you so much for all of the information. We truly appreciate you giving your time to the patients and to the rest of the community. Thank you all, and good night.
Good night.
Thank you. Take care.
Thanks.